First of all, progress on the LBC 1936 project. Well, many of you will know this, but I'll just introduce quickly for those of you who don't. On Wednesday, the 4th of June 1947, Scotland tested its entire nation's intelligence. 70,805 children took the Murray House test number 12 in a national IQ test, which tested about 95% of people born in 1936. So if you're born in 1936 and you were at school in Scotland in 1947, you pretty much got this IQ test. And the Lothian Birth Cohort project of 1936 follows people up who took this IQ test at age 11 in Scotland in 1947. And these are the data that we start with as our baseline, the data from the Scottish Mental Survey of 1947, which has got validated IQ data for pretty much the whole of Scotland born in 1936. And this is what one of our subjects looks like, and our subjects do let us show pictures of them at these conferences. And so this chap was here, aged 11 in 1947, when he took the Murray House test number 12, and he was followed up by the Lothian Birth Cohort's 1936 team at age 70 in wave 1 of the follow-up, and it shows you the years there, at age 73 at wave 2, and now we're just putting into the field wave 3 of the project, which will study people at average age 76, and that will run between 2011 and 2013. So far in the Lothian Birth Cohort 1936 project, what we've done is the following. We've got our baseline data from age 11, and at age 70, or average age 70, we've got mental test data and also collected data at age 73. And down there, it shows you also that we have genome-wide SNP data on the Lothian Birth Cohort, and also at age 73, they've all had a structural MR brain scan that I'll tell you more about. So we now have data at age 70 and age 73. Now, although we all know that between age 70 and age 73, it's not a long time, one's not expecting much change over this relatively short period, the fact is the only way to do a third and a fourth follow-up is actually to have done a second in between times. So we are going to present data from age 70 to 73 today, and Wendy Johnson's going to do that, but we're not expecting much in that short period. As I say, the age 73 is a staging post for later, more interesting follow-up waves. And that, in fact, that wave is funded by Age UK, who also funded the two previous waves, and we're just getting going on this now. So, what data have we been collecting on the Lothian Birth Cohort of 1936? Well, as I said earlier, they all took part in the Scottish Mental Survey of 1947. They live in the Edinburgh area, and I've often regretted not calling it the Edinburgh Birth Cohort, which would have been a lot simpler, but the Lothians, the Edinburgh area. 1,091 were tested at wave 1 of the follow-up between 2004 and 7, and these are relatively healthy individuals who are screened for dementia, both using mini-mental state and medical history. For those of you who would be interested in the cognitive assessment, they go through a large battery of cognitive tests, including sitting the same test again as they took at age 11, the Murray House test number 12 that was done at age 70. They have a number, those of you who know my work know I'm interested in processing speed especially, so there's a lot of tests of processing speed, quite a few tests of memory, as is listed there, and beyond that, matrix reasoning, verbal fluency, and block design. And we also do the National Adult Reading Test and the Wexler Test of Adult Reading as well. There are lots of other things too, though, social and biomedical assessments and a long questionnaire. And so when they come and do the cognitive tests at our Wellcome Trust Clinical Research Facility, they also have a social and medical history taken by the testers. They report their mood states, and we do the mini-mental state examination, a detailed physical exam that's listed, some of it's listed there. We take blood for an increasing number of biomarkers, and we cheat by getting an extra few hours of work from them by asking them to bring along a questionnaire completed to the clinical situation that they've already filled in at home because we posted it in advance. And these are some of the things that we do in the questionnaire. So, at wave 2, when we saw them at mean age 73, we did the same assessments as at wave 1, which is at age 70. But by that time, we had extra funding to add in a structural magnetic resonance brain imaging scan, the scanning of the big arteries in the neck, the carotid arteries using Doppler ultrasound, photographs of the retinal vessels at the back of the eye, which is the only place you can see the brain's blood vessels in the body, genome-wide association using the Illumina 610 chip. That is, we did over 600,000 single nucleotide polymorphisms on them, extended the biomarker assessments, and extended the well-being assessments. So at wave 2, out of the 1,091 that were tested at wave 1, we saw 866 and documented from the others why they didn't show up, whether that was death, relatively few, dropout for other reasons, including medical problems. 821 of those had a full carotid Doppler ultrasound scan, and 728 of them had a brain scan initiated. And at wave 3, which is just kicking off any day now, Disconnected Mind, again, it's part of the Age UK's Disconnected Mind project. It'll run that project between 2011 and 15, and once again it'll have clinical cognitive assessments and repeated brain imaging. So by that time we'll have 2 brain imaging assessments. So let me tell you a bit about the collaborative network, because as it's grown and we've brought in more things that we analysed in the Lothian Birth Cohort 1936, we've accrued a set of collaborators. So first of all, collaborators in the actual team that exists funded in the Lothian Birth Cohort. I'm going to start from 1 o'clock here and work round. So Alan Gow works mostly on engagement matters in the project. Janie Corley, I'm going to be fairly brief here, works on lifestyle contributions to cognitive ageing. We've got a number of geneticists, Sarah Harris, Michelle Luciano, Lorna Lopez, Gail Davis and Andy McLeod. Renee Motis works on personality, quality of life and health. And on imaging, Lars Penke, who's talking today, Tom Booth's coming to the team, Benjamin Aribisala, Maria Hernandez-Valles and Susana Maniega work on imaging. And those of you who know Wendy Johnson won't be surprised to say that she pretty much works on everything in the Lothian Birth Cohorts of 1936. It's hard to get a smile out of Wendy, actually, which hasn't happened, actually, I can see. Yeah. And so those are the talks you're going to hear today. Lars is going to talk about some of the brain imaging. Alan's going to talk about engagement. And Wendy is going to talk about the follow-up from 70 to 73. And unfortunately, Gail Davies, who's actually as a customer at Edinburgh's New Royal Infirmary this morning, I'm actually going to give her talk for her. And outside of the narrow team, but within the University of Edinburgh, we have a number of collaborators as well. All our testing is done at this wonderful Wellcome Trust clinical research facility, where we do our clinical testing, genetics and brain imaging. We've got excellent collaboration from geriatric medicine, from human genetics, from brain imaging, from mood imaging and genetics in the Department of Psychiatry, from ophthalmology, oxidative stress in the respiratory medicine department, and from cortisol studies in the geriatric medicine and endocrinology departments as well. So we've got a good set of collaborators within the University of Edinburgh. But even beyond that, we've got collaborators who contribute to specific areas of study within the Lothian Birth Cohort of 1936. So with regard to diet and nutrition, we work with the Rowett Research Institute in Aberdeen. We have specialists on biomarkers through in Glasgow, specialists on brain imaging, blood-brain barrier biomarkers, on telomeres, immunology, proteomics, genome-wide studies, and brain cortical thickness. So all of these are collaborators outside the University of Edinburgh. And also, like everybody else who's got genome-wide association data these days, we're involved in a large number of genome-wide association consortia. And this slide from Lorna Lopez and the team has actually sorted it into 3 groups. Those genome-wide consortia based on body phenotypes, those in the middle based on brain and psychology phenotypes, and those at the right based on blood phenotypes. And so you can imagine members of the team actually spend quite a lot of time these days on teleconferences and international calls. It makes for a slightly odd world whereby last week, the team had 2 Nature Genetics articles accepted as part of these large consortia, and it's a very interesting thing to have to negotiate. So some recent findings, and these are findings that won't be presented by the other speakers this morning. So, for example, if you look at Alan Gow's recent paper, one of the things we've been able to show using both Lothian Birth Cohorts, the 1921, which I'm not going to talk about in detail this morning at all, and the 1936, we now have the stability across the life course of the Murray House test number 12 test of intelligence. And so at the top, you can see that the Murray House test number 11, given to Lothian Birth Cohort of 1921, we have a correlation between age 11 and age 87 now. It still holds up at 0.51. And for the Lothian birth cohort 1936, if you look at the bottom, the raw correlation is 0.67 between age 11 and age 70. But if you disattenuate that for the restricted range of those individuals that we see at age 70 by comparison with how they were at age 11, the correlation goes up to 0.78, which shows a rather interesting thing, that at least half the variation in intelligence is stable from age 11 into old age. But of course, it's the other half we're particularly interested in. What we want to try and explain is the change in mental ability from childhood to old age, and what are the determinants, what makes people differ, i.e. especially be more successful in cognitive aging than you would expect from their mental ability at age 11. Let me show you some results. First of all, one thing that we've made rather a specialism of is reverse causation. That is, it's quite common in cognitive aging to find a variable that's associated with cognitive function in old age. But the thing we can then ask is whether, in fact, individual differences in cognitive ability at age 11 actually caused potentially some of the variation in that particular variable in old age, and that's called reverse causation. Of course, it could also be because of confounding, whereby a set of variables is causing individual differences in both the cognitive outcome, bless you Ted, and also the variable that's meant to be contributing to the cognitive outcome. And let me just show you some of those. And this is what Janie Corley and the team has made rather a speciality of in the last few years. She's shown that for caffeine, body mass index, and alcohol, in each case, drinking more coffee, drinking more alcohol, and being lighter in body mass index are all associated with better cognitive function in old age. Yes, you're smiling, but rather too quickly. So I'm about to pull the rug from under those particular feet. Because once you then bring cognitive ability at age 11 into the model, none of those associations is any longer significant. So the story is it's bright children who end up drinking posh coffee, it's bright children who end up drinking red wine, and it's bright children who end up being lighter in old age rather than heavier. And it's not about these things causing better or worse cognitive aging. That's not the case for smoking. It's the case that brighter children don't smoke, and brighter children tend to give up smoking, but smoking over and above that reverse causation effect also causes deleterious cognitive aging as well. And again, I'm just showing that that's one particular area that Janie has specialised in the group. Another thing that we've published recently, this is Wendy Johnson's work, is looking at the hierarchical structure of intelligence. This is the earliest diagram I can actually find of the hierarchical model, and it's quite nice because it was done by P.E. Vernon when he was in Glasgow in the 1940s. And this is the idea that Carol later has absolutely stamped on all of our foreheads, that there's a general factor, then domains of cognitive function, then test-specific variation. And we've, in the Lothian birth cohort 1936, been rather careful to try and get these domains reasonably well represented in the battery of cognitive functions. And Wendy's just had a paper accepted in Intelligence looking to see whether the Lothian birth cohort data actually fit her Johnson and Bouchard VPR model of the hierarchical structure of intelligence. Those of you, again, who know that the thing that kind of obsesses me is that I'm particularly interested in speed of processing, and we have 5 different speed of processing tests in the Lothian birth cohort 1936. But we found again a rather interesting set of potential reverse causation there, because speed of processing, as you'll know from the work of likes of Tim Salthouse, is the quintessential or exquisite variable in old age. It's meant to be the marker of how the brain is doing in old age. And sure enough, if you look at the R-squared with Murray House test at age 70, each of these 5 markers of information processing is indeed significantly associated with Murray House test intelligence, inspection time, reaction time, and symbol search and digit symbol. However, if you adjust that R-squared for the Murray House test taken 60 years before, that's what happens to the R-squared. And again, it's quite early in the morning, so I'll stick the attenuations in for you. You don't have to look at it, but look at those numbers at the right-hand side. Those are enormous attenuations. So how quick your brain is at age 70 is not a state measure. It's a lot of that is to do with how bright you were 60 years prior to that as well. And so the conclusion is that brain processing speed is often used as an index of current state of brain performance. But as it says there, individual differences in processing speed at age 70 are most heavily dependent upon general intelligence at age 11, 60 years earlier. Another thing we've looked at, just to pick out a few things we've got in press recently, is telomeres And those of you who don't know, telomeres are the bed socks that sit at the end of your chromosomes, so that each time your cell divides, it doesn't actually ribble out. Ribble, Scottish word. You know the way a loose thread comes out in a cardigan and starts pulling it all out? Well, that's what ribbling out means, so that your chromosomes don't start ribbling out. And in fact, these get shorter each time your cell divides, but they also get shorter because of oxidative stress. That is, how rubbish your environment is actually affects your telomere length. And of course, at one time, me as well as lots of other people thought that telomere length was going to be the great representative of how much the environment had rubbed your brain away during your life course. Well, in fact, it turned out to be a bit of a washout. In fact, these are all the variables that we've related telomere length to in the Lothian Birth Cohort 1936. Now, there are publications on every one of these areas in telomere length. That is cognition, social background, biomarkers, and fitness. And I said to Sarah Harris, who's the lead author on this, let's not bother trying to answer all of these areas. Let's just put it in as one null finding. In fact, that's largely what we found, because these are the significant findings, that in women alone, telomere length, that is longer telomere length, is associated with lower levels of the immunological biomarker C-reactive protein and better fluid intelligence. So longer telomeres have got these sensible associations in women with these, but that is two associations in quite a large number and could well be type 1 errors. They've not turned out to be the marker of environmental oxidative stress as one would have hoped. We've looked at personality in the Lothian Birth Cohort of 1936, and Renee Motus will be talking about that in another talk, not in this session. We've looked at quality of life. Cognition is not the only important thing about old age, and we look at quality of life as well, and we've been publishing on the determinants of that. And also we've been looking at health literacy. It seems to be obsessed in the US just now. Health literacy seems to be absolutely everywhere. And one nice thing we were able to do is take 3 current tests of health literacy. I'm not going to go into these in detail, and Catherine Murray is the lead author on this. The S-TOFLA, the Realm, and the newest vital signs. And we're able to look at these at age 70 and find out, in fact, that these have got strong associations with age 11 IQ. And when you look at current cognitive function and education, and whether these, in fact, make contributions to health literacy above and beyond age 11 IQ, there are some relatively small effects. In one case, the IQ change from age 11 to age 72 is quite a big effect on the S-TOFLA. But a lot of what people are looking at with regard to health literacy, once again, as a state measure that affects one's ability to handle health data, is in fact traceable back to cognitive function 60 years earlier. We've also done what we think is the first proteomics study of cognitive function, that is looking at the urine of the Lothian Birth Cohort 1936 and measuring the 1,500 or so peptides that exist there to see if there are peptide profiles that differ between people of high and low intelligence. Although this is purely a pilot study, we took 20 people from the very top and 20 people from the very bottom. It's expensive and extremely burdensome to do urinary proteomics. We found some indicative proteins that differ in the urine between people of high mental ability and low mental ability, and we are now upscaling this pilot project to do a full project. We've collected urine on all of the, or pretty much all of the Lothian Birth Cohort 1936, and plan to upscale that to see if that's in fact a more proximal biomarker than looking at genetics. One thing recently we've looked at in our collaboration with McGill in Montreal, done by Sharif Karama in the Alan Evans group, is looking at the brain cortical thickness. And just double checking, Lars, you're not presenting this, are you? No, good. Right. Sharif Karama has just said that we can actually show these slides because they're just very new data. Sharif has measured, if you look at me just for a second, the cortical thickness, the grey matter thickness around the brain of the Lothian Birth Cohort 1936. And that's the pattern whereby the correlation between IQ at age 70 and the thickness of the cerebral cortex. And those of you who are thinking P-fit are right. This is not a bad fit to the P-fit. But as you'd now expect me to do, let me show you the correlation between age 11 IQ and cortical thickness at age 70. I'll repeat that. The next slide will show you the correlation between age 11 intelligence and cortical thickness from age 70. And this again, the N here, I'm not got exactly, it's about 500. And that's it. It's also reasonably impressive. So let's just look at this slide. the cortical thickness at age 70 and intelligence at age 70, and then take out of that IQ at age 11. And that's what's left. And that area that's still there, that is cluster-wise significant, is actually really interesting with regard to potentially cognitive aging as well. So you have two nice stories there about reverse causation or confounding, and on the other hand, about cognitive aging. Now, at that point, I'm going to thank the Lothian Birth Cohort team, Age UK, who are the foundational supporters of the Lothian Birth Cohort 1936, and other ancillary funders. Medical Research Council fund the imaging, the brain imaging analysis, and BBSRC funded the genome-wide association studies. So thanks very much. That's the Lothian Birth Cohort 1936.